Hi everyone, today we're going to look into the 3 other processes of material absorption through the cell membrane and into the cell. Now first we're going to start with active transport here. Now active transport, it differs from diffusion in the sense that it requires energy in the form of ATP in order to transport molecules from an area of low concentration to an area of high concentration. So essentially against the concentration gradient. So let's kind of write it down here. So it uses energy, and it uses energy in the form of ATP, which is then converted into ADP plus a phosphate group, which produces energy in the process. And this energy is used to transport molecules from an area of low concentration to high concentration, which wouldn't exactly be possible if it were to be diffusion, because diffusion essentially under the Brownian motion, the molecules would move from an area of high concentration to low concentration due to their random motions. Now, an example of this active transport in application is the sodium-potassium pump, which maintains a proper concentration of sodium and potassium ions inside and outside the cell. So let's quickly draw the pump here. So what this sodium-potassium pump does is essentially transport every 3 sodium ions outside of the cell for every 2 potassium ions that are absorbed into the cell for every ATP molecule that is consumed. So what happens is, let's say we have sodium ions here, potassium ions here. For every 3 sodium ion that is moved outside of the cell, transported outside of the cell, it transports inwards every 2 potassium ions, and this is for each ATP molecule that is consumed or used. Now, this is crucial for maintaining the resting membrane potential, and a case of its application is neurons in conducting nerve impulses. Now, next we're going to look into endocytosis. Now, endocytosis is a process by which the cell engulfs large particles or fluids from its environment, and this occurs as the plasma membrane invaginates, i.e. folds inwards, and forms a vesicle that contains the ingested material. So I'm just going to visualize everything for you. Say that this is the substance to be ingested. What would happen is, if this were to be the membrane, and this is the substance, the membrane would essentially fold inward or invaginate. And what would happen after that is the substance that's ingested would be contained in a vesicle like this, pretty much. And that is endocytosis. Now, endocytosis can essentially be classified to 2 main types, and this is phagocytosis and pinocytosis. Now, these two differ in the sense that phagocytosis is where the cell eats or engulfs large particles like bacteria or dead cells. And we looked at this earlier on when investigating the endophagocytosis theory, when looking at the case of mitochondria, or the cases of endosymbiosis and chloroplasts, and how they actually evolved to be inside eukaryotic cells as organelles. And in the case of pinocytosis, this happens when a cell drinks extracellular fluids or even dissolved solutes. So this is essentially liquid endocytosis. I guess that's a more intuitive expression of this specific type of endocytosis. Right, and the last process that we're going to look at is exocytosis. And this is kind of like the reverse process of endocytosis. And what happens is, it's when cells kind of try to expel substances that they do not need, such as waste products. And it happens in a process where vesicles kind of fuse with the plasma membrane. So when we were talking about endocytosis, we learned that these substances that want to kind of be engulfed by the cell are contained within these vesicles. Right, but in the case of exocytosis, because these substances want to be expelled, the vesicles essentially fuse with the plasma membrane. And as it fuses, the materials that are contained within these vesicles are released into the extracellular environment and thus dispelled from the cell. Now, exocytosis is an important process for processes like even neurotransmitter releases from nerve cells and the secretion of insulin from pancreatic cells, and those cases are specific applications of exocytosis in our bodies. right? And that kind of brings us to the end of this video. Thank you for watching, and I hope you have a better understanding of the different methods used to absorb or release materials from cells through the cell membrane.